Most of us humans surrender to the forces of nature, and honestly we're one of the most curious creatures in this giant floating ball we call Earth. Our ultimate gift to face the struggles of this life is our brains. Now imagine if you are able to transition from intelligence to a hyper-intelligent super being that will defy the limiting form of a human, or having a technology that could probably cure all diseases. Well, that's close to being real more than ever with the latest discoveries on gene editing. As utopic as it sounds, these latest discoveries have their dark side. So join us today for a quick look at how gene editing can change the world. But before we start, make sure to subscribe and hit that bell to turn on notifications. To change an organism's DNA, whether a human, shark, or the broccoli served on your table, geneticists and scientists spent years developing a group of technologies called genome editing. These technologies allow genetic material to be added, removed, or altered at particular locations in the genome. During the years, several approaches to these technologies have been developed, but the one that stands out is known as CRISPR-Cas9, which is short for clustered, regularly interspaced short palindromic repeats and CRISPR-associated protein 9. I know, forget my gibberish. Here's a practical and yet a simple explanation of this technology. CRISPR-Cas9 is a unique technology that enables geneticists and medical researchers to edit parts of the genome by removing, adding, or altering sections of the DNA sequence. The CRISPR-Cas9 system consists of two key molecules that introduce a mutation into the DNA. The first one is an enzyme called Cas9. This particular enzyme acts as a pair of molecular scissors that can cut the two strands of DNA at a specific location in the genome, so that bits of DNA can then be added or removed. The second one is a piece of RNA, which is a nucleic acid similar in structure and properties to DNA, but it only has a single strand of bases. Anyway, an RNA main job is simply guiding Cas9 to the right part of the genome. This makes sure that the Cas9 enzyme cuts at the right points in the genome. At this stage, the cell recognizes that the DNA is damaged, and that's because some bacteria have a similar built-in gene editing system to the CRISPR-Cas9 system that they use to respond to invading pathogens like viruses, much like an immune system. Using CRISPR, the bacteria snip out parts of the virus DNA and keep a bit of it behind to help them recognize and defend against the virus next time it attacks. This technology has a great potential as a tool for treating a range of medical conditions that have a genetic component, including cancer, hepatitis B, and high cholesterol. The discovery of CRISPR-Cas9 was in 2011 by French professor and researcher in microbiology, genetics, and biochemistry Emmanuelle Charpentier. She discovered an additional key component of the CRISPR system, an RNA molecule that is involved in recognizing bacteriophages, which are an abundant type of virus that infect bacteria and other one-cell organisms. They inject their own DNA into a host cell, hijacking the host cell to copy their own DNA and make more phages. A year later, she teamed up with American biochemist Jennifer Doudna, who both won a Nobel Prize for their discovery, which is again the ability to simply isolate the components of the CRISPR-Cas9 system and adapted them to function in the test tube, which proved that the system could be programmed to cut specific cuts in any DNA sequence desired. This golden discovery is not confined to medicine only. The CRISPR technology is applied in the food and farming industries to engineer probiotics cultures, which are part fermented dairy products like yogurts against infections. It is also being used in crops to enhance yield, drought tolerance, and nutritional value. And in the future, the CRISPR gene editing could potentially be used to create new species or revive extinct animals from closely related ones. In November 2018, He Jiankui was the first person to use CRISPR-Cas9 on humans. He claims that he had created the first human genetically edited babies, Lulu and Nana, and that's by editing the embryo's genomes to disable a gene called CCR5, which encodes a protein that allows HIV to enter cells. He was aiming to mimic a mutation that exists in about 10% of Europeans and helps to protect them from HIV infection. But he might have inadvertently caused mutations in other parts of the genome which could have unpredictable health consequences. There's lots of ethical issues with Jiankui's experiment. Firstly, The clinical project was conducted secretly until 25 November 2018, when MIT Technology Review broke the story of the human experiment based on information from the Chinese clinical trials registry. Compelled by this situation, he immediately announced the birth of 2 healthy genome-edited babies in a series of 5 YouTube videos the same day. Secondly, the common scientific practice should have been a set of tests of these mutations in an animal model to measure the unexpected effects of the experiment. This is probably the most upsetting part of the experiment. The 2 babies were test subjects for a genetic mutation that scientists didn't have time and care to test in animals and study the short and long-term impact. On the 30th of December 2019, Chinese authorities announced that Dr. Jiang Kui was found guilty of forging documents and unethical conduct. He was sentenced to 3 years in prison with a $430,000 fine. Despite this reckless and unethical use of the technology, CRISPR-Cas9 will probably become the elixir of life the human race was always seeking, and soon enough, it could save thousands of patients waiting for donated organs. The animal organ, probably from a pig or a baboon, could be genetically altered with human genes to trick a patient's immune system into accepting it as a part of its own body. The lack of organs available and the constant battle to keep immune systems from rejecting allotransplants will become from the past. With that being said, we made it to the end of the video. Let us know your thoughts in the comments, and if you like the video, make sure to subscribe and hit the bell to turn on notifications. I'll see you in the next video. Take care.